Now I would like to introduce Julie Douville, who will take over and see the whole oversight of discovery and development.
Good morning. Thank you, Nadina. That's me. All right, so now we're going to cover how we take the ASOs that the team in the lab are screening and discovering, and how do we move them all the way through an IND submission and treating a patient. And I'll discuss as briefly as possible the different processes that are supporting this transition, and then we'll move into our regulatory processes and status. So for the non-clinical development steps, you need to have a lot of different skills and knowledge, and this team is bringing exactly that into n-Lorem. And you can see we have a toxicologist, a chemist, we have Nadina, myself, and Stan. And so taking all of our experience, expertise, knowledge together really makes it possible to continue the transition of these ASOs. So Nadina covered the first few steps here, and so then I'll take you through the rest, where we run the tolerability study, which is the last step of the screening, and then to lead into a data package that will be presented, reviewed at the RMC committee to select a lead ASO, moving into the more critical steps of development, which include GLP tox and manufacturing, and then into the IND submission and approval. So the non-clinical team is involved in many critical roles within n-Lorem. ATTC, RMC, the management of our partners, CROs and CMOs, as well as the regulatory efforts. The RMC committee, Nadina talked about it. It has many roles, but more importantly, it is where we select the ASO from all the ASOs that were designed and screened. Which one do we take into development to then go to the clinic? And so we have external experts on this committee, as well as internal experts at n-Lorem, reviewing all the data to make sure that we pick the best one. The RMC committee will also discuss the challenging cases, as Nadina explained, and then we also use this forum to discuss other challenges that we encounter, such as how to best optimize our CROs and CMOs, and how we could better standardize the data collection, for example. But we can also bring up any other topic that is relevant for the organization at that moment in time. And now I want to really focus on how we take these beautiful ASOs that Nadina screened in the lab, and how do we turn them into a drug that will treat our patient. And the next steps involve working with our partners. So those are the CROs and CMOs that I mentioned. CROs is contract research organization, CMO contract manufacturing organization. And so essentially what we do at this step is to assess the safety of our ASOs, mainly through the animal studies, and then also manufacturing the ASO with rigorous criteria to make sure it's adequate for human administration. And the overarching goals of this step is always to move things faster and more efficiently so that we can bring an ASO sooner to our patient. So at the core of this is really the collaboration with the contract organization to be able to increase efficiency. And so we have partnered with CROs and CMOs that have years and years and years of experience working with the biotech industry, large pharma, including Ionis. And so we leverage this experience to be able to maximize efficiency and reduce cost. And that doesn't mean that our team isn't involved. On the contrary, our team oversees all the activities that are conducted at the CROs and CMOs, and this allows us to mitigate the risk and errors. It also ensures that we stick to the timeline. We are also able to ensure that the study will be conducted according to protocol, and we're able to identify issues proactively and also address them. So at that stage, it's really important to build redundancy. The CROs, CMOs, they work, as I mentioned, with many other companies, biotech, large pharma, and so they're very busy. The work that we do with them also is quite specialized, and it is not an easy thing to schedule. And so that's why we have this incredible network, which really allows us to have the best pricing, but also the best schedule, which is important for our patient. The tolerability study, so I just briefly wanted to mention what it is the last step of the screening where we put our best ASO into rodents, and then we look at a short battery of endpoints that are really critical, and this allows us to identify any ASO that would be problematic. And so if ASOs are not meeting the criteria, they don't move forward. And so this is ultimately what allows us to pick our best ASO. And then GLP tox is a regulated study that is really important in the IND filing. And we use this study to identify what are the potential toxicity of the ASO, what are the target organs, and that sort of thing. And so typically this will be a repeat dose study. So animals are dosed every month, similar to our patient in the clinic. And usually lasts 3 months. And we look at a more extensive battery of endpoint in that study compared to the tolerability. Then for manufacturing, there are 2 steps. The first step is to make the drug substance. So this is the ASO in a powder form, and essentially taking those building blocks, the ATCGs, and then putting them in the right order so that we can bind the target that we want. And there are strict criteria when making this drug substance to make sure that the resulting drug will be adequate for treating a patient. And this step is interesting. The smallest batch that we can do under the GMP regulation will give us enough drug to treat a single patient for 10 years or even more. The second step of manufacturing is making the drug product. So this is taking the powder form of the ASO, formulating it in a liquid form, which can then be administered to the patient by the hospital. Once again, there are strict criteria for purity, sterility, all of that to make sure that the drug is safe to administer to human. And at the end of these steps, we will end up with 200 to 300 vials of our ASO. I made this slide here to show you how we have all these processes running in parallel to make a very effective timeline and not have any gaps between the steps, essentially. So while the toxicology is running, manufacturing is going, and we're also working on the IND. Now, this doesn't happen just by picking up the phone and asking our CRO, can you run a study? There's a lot of activity that happens in the background to make this happen. And so this is what I'm trying to illustrate here. So our lab will screen ASOs and hand over a list of ASOs to the non-clinical development team. And then the team will then order the synthesis of these ASOs to one of our partners. Once the synthesis is completed, the ASOs will be shipped back to our lab, and our lab will formulate these ASOs and then ship the formulated ASO to the CRO that is running our tolerability study. So animals will be dosed, and the CRO will collect tissues and send the tissues back to our lab for analysis. Now, when the lab has analyzed the tissues, they will communicate the result to the non-clinical development team, and then the team will look and analyze these results and then put all that together in a data package that will go to RMC to select the lead ASO. Then once we have a lead ASO, the non-clinical team will once again order the synthesis of the ASO with one of our partners, and then ASOs will be shipped to the CRO for the toxicology study. Animals will be dosed. Data will be collected, analyzed, put together in a report that will be sent to the team. And then along with the report generated from the tolerability study, we will take the GLP tox report, put that together to populate the relevant section of the IND. So this is what happened for one program. So you can imagine if we have 2, 3, 4, 5, 6 programs running in parallel at different stages, how complex this movement can become. And similarly for manufacturing, when we have a lead ASO, this is communicated to our team, and then our team will contact our partner for drug substance manufacturing. The ASO will be synthesized, purified, all of that. The data will be sent back to our team for review and analysis. And then at the same time, our team will contact our drug product manufacturing partner and say, hey, we have ASOs coming your way. And then this CMO will formulate the ASO in a liquid form, once again, send the data to our lab for review and analysis. And then our team will put all of this data together and write the relevant section that will go in the IND. And at the same time, the drug product CMO will ship all of those vials to a different partner. They are called a third-party logistic vendor, and their role is to essentially store our vials under the appropriate conditions so that the drug stays stable and good for a long time. And so then once we have an approved IND, the team will communicate with the third-party logistic vendor to say, hey, can you ship 50 vials to this hospital where our patient is going to be treated? And then throughout the treatment period, there's constant communication between the treating institution, our team, and the inventory storage facility to ensure that we have a steady supply of drug at the site where our patients are dosed. So it's really important to have good partners in order to have the best possible cost. So this slide here shows you the cost of an average program and the cost, what each piece of the program, how much it contributes to the overall cost. And so with our partners giving us generous discount, free batches, all sorts of good stuff, well, we are able to bring this cost down by 40%, 40% cost reduction, which is really important. It allows us to, develop more ASO for more patients. Here, I wanted to also highlight another aspect of this partnering, and it's the scheduling part of it, the timeline. This is so critical for so many of our patients. So here is an example of our toxicology schedule for a given year, where you can see at the beginning of the year we have program A that has started, program B that has also started. Both programs are going very well in the GLP tox. However, at some point we get data from our lead ASO on program B that makes us question whether this was really the best ASO and is it still appropriate to continue with this ASO. So we don't want to waste any time, and so we call our partner, CRO, and we say, hey, would you be able to schedule another study real quick so that we can also test our backup ASO? And so they said, yeah, we can do that. And as you can see, they give us a slot that is quite interesting from a timeline perspective. So we're happy. But then we get new data that confirm actually that our lead ASO was really good and that we can continue to move forward with that ASO. So that's great. However, now we have an empty slot and we need to figure out what to do with it. We look at our portfolio and then we realize that the program C is ready to go. We have the ASO is available. It's been synthesized already. So it's like, great, we can move this program up and use this empty slot. This has just moved the problem, as you can see. And so can we do the same with program B? Unfortunately, the ASO is not ready. We don't have it yet. So we call our partner CMO and we say, hey, would you be able to make an ASO as quickly as possible and make us able to move this program up? And so our partner said, yes, we can do that. And so this was a really good news. And so we were able to move program D up and then subsequently move the entire portfolio up and be able to run, if you have seen what happened there, we've been able to add an additional program to the portfolio within the same period of time. So that was really important to also mention. There's the cost, but also the timeline that is important. So we're going to move into regulatory activities now. So you've heard IND many times. What is it? Sarah explained it. It's an investigational new drug application. So this is essentially the big package that we send to the FDA. It tells the whole story of our ASO, how we made it, what it is, what it does, and the story of our patient as well. Who's the patient? What's the disease? What's the target gene? What's the mutation? We do research INDs, and so the research INDs, they have different sections that I will cover in the next few slides. So the first module is an administrative module. There's not a lot to say here. It has a cover letter. It has the general investigational plan, which is a summary of the entire IND, essentially. It also includes some forms, because that's, the FDA. And the CV of our principal investigator to show that our physician is capable of running this research study. And then there's a CMC module, so chemistry, manufacturing, controls. So this is the manufacturing. What is the drug? What's the sequence of it? How does the molecule look? How was it synthesized, purified, manufactured? It has all the results of all the tests that we put the drug through. Is it, pure and free of contaminant and all of that? And then we also put for both the drug substance and the drug product, we also have a section on diluent that we use when we formulate the drug at the hospital to treat the patient. We include all the certificate of analysis, of course. It has the actual label that goes on the vial is included in the IND. And we also prepare a pharmacy manual for each patient, for each program, so that the hospital know how to prepare the drug. There's a non-clinical module in which we put the animal studies, but also all the in vitro testing that the ASO went through. And so there's the actual study reports, but also an overview which puts all the data together and summarizes all of that. And then finally, there's a clinical module. So the clinical module will have the clinical protocol, so the actual protocol that our investigator will use to treat the patient. It details the dose level, escalation, the dosing interval, and what endpoints we'll be measuring. It has the informed consent and a clinical overview, which will talk about the patient-specific mutation and phenotype, the disease, the biological effect of the mutation, and how the ASO will act on this target to essentially address the root cause of the disease. We'll also discuss the various treatment goals, outcome measure, and the risk-benefit for each patient. So once we submit this, what happens? So the FDA can ask for clarification or additional information on any aspect of the submitted IND. And oftentimes, they will request a response very quickly. The FDA has 30 days from the time we submit to give us a response on the application. And so that's why this turnaround time is very critical. We can have 3 possible responses to our application. We can have an approval to proceed, which is, of course, the best news. Or we can have a clinical hold. The clinical hold can be partial, so that means they will allow us to proceed, but they will limit the dose at which we can go. So they'll say you can go, but no more than 20 mg, for example. And then we can provide more data or information to then convince the FDA to let us go higher if it's appropriate for the patient. We can also end up with a clinical hold for various reasons if the FDA has any concern. But then again, this is a situation that we can resolve by providing clarification, additional information, or simply discussing with the FDA. So we've been able to really streamline this process. We have a good oversight on the timeline. We have now good processes so that the various experts within n-Lorem can prepare the different sections of the IND in a coordinated fashion. We're able to timely react to any FDA request, and we've been applying the learning. So when the FDA tells us something on one program, we're able to rapidly, pivot and apply this learning to the next IND so that we don't have the same comment from the FDA. And so the past few applications were really streamlined, and we essentially had no questions or comment from the FDA. So I'm going to go over a few metrics now for the regulatory aspect. So we have had over 130 regulatory interaction with the FDA so far. We've had 5 formal pre-IND meetings with the FDA. So this is where you discuss early on with the FDA to present them your plan, and they can provide advice at this point. We've also had informal meetings with the FDA. This is a bit unusual. But we've been able to use this forum to discuss different challenges, including moving beyond N of 1, and I'll cover that in the next slide. So far, as Sarah mentioned, we've submitted 21 INDs in about 2 years. All of these INDs have been approved to proceed. And these 21 INDs are covering 29 patients. And so I wanted to touch on moving beyond N of 1. So there is, maybe some of you are aware of that, there's a regulatory gap between a commercial program where the industry has very specific rules that they need to follow to get an approval to commercialize a drug. And we have the N of 1 guidelines, or the individualized ASO guidelines, but there's nothing in between to address 5, 10 patients. What do you do with that? And so there's different reasons why we may want to move an N of 1 to an N of few or N of many. It could be that the known prevalence of a mutation increases as whole genome sequencing becomes more available. And can also be other reason. We can change the strategy, which makes the ASO, if an ASO is non-allele specific, for example, then more patients can use it. And so as we prepared more and more INDs, we gained knowledge, experience, and we learned that if we already had an IND for an ASO, we can leverage documents that were already submitted to really be able to streamline the next IND. And then we also learned that if a small number of patients have the same mutation, similar phenotype, it can be possible to group them under a single IND. And so this is really how we've been able to industrialize this process to be also able to increase efficiencies. And so using this strategy, we now have 5 different ASOs that are approved to treat between 2 and 9 patients each. And so I'm going to do the math for you here. We have 15 ASOs. For most of these ASO, we filed 1 IND to treat 1 patient. And then for a subset of these ASOs, we filed a second IND for a second patient. And then we have this IND here, for which we filed this ASO for which we filed 1 IND to treat 3 patients. And then just at the bottom here, for this ASO, we've been able to file an additional IND, a third IND, to treat 7 more patients. So I thought that was really cool. So to conclude, I hope I showed you how we have industrialized every step of the process from discovery to regulatory approval, and that we have established very efficient processes that are tailored to our specific needs at n-Lorem for our patient, and that our team of experts is really moving these ASOs as quickly as possible from the lab to the clinic, and that, of course, we could not do that without our partners from the industry. And so all of this means more ASOs for more patients more rapidly, and then more knowledge that can be applied into the process to make it even better. So thank you for your attention. I think I have a slide showing all of our fantastic partners. So thank you, and thanks to our partner, and you can also thank our partners.